I'm Kevin Lawson. I'm the VP of Regulatory Affairs. IMMCO was founded in the early 70s as an offshoot from UB. Some of the specialists there saw a need for autoimmune disease testing, and it started with skin antibodies. It quickly grew into other kinds of autoantibodies as immunology and autoimmunity became more and more well known. And since then, we've found that a number of diseases, diabetes, connective tissue diseases like lupus, celiac disease, all involve autoimmunity. IMMCO has developed a number of kits to identify antibodies connected with autoimmunity. IMMCO manufactures between 50 and 200 different kinds of kits, depending on whether you're talking about research use kits or in vitro diagnostic kits. This is the ELISA validation laboratory. We have a few pieces of equipment where we're validating the use of our tests on these particular devices, and these tests are going to be used for FDA 510K studies in support of the instruments. This is the assay development laboratory. Our research scientists are developing next generation ELISAs as well as new technologies like microarray proteomic assays for identifying antibodies that are present connected with a variety of autoimmune diseases. The R&D scientists have to put it through its paces, test it. We need to show to FDA the safety and efficacy of these assays. So they'll do initial feasibility studies. They'll optimize the assays for use in the laboratory and with equipment like we've seen in other areas of the building. We'll test a variety of patient samples, clinical studies, etc. And once we have all of that information together, we'll submit to FDA. And that process takes anywhere from 3 months to a year. This is the cryosection laboratory. These cryostats are used to cut very thin sections of tissues so that we can see antibodies bind onto the tissue sections. The tissue sections are adhered to glass slides, and the glass slides are observed under a fluorescence microscope.
So this is our clean room here. In here we do a lot of our ELISA plate making. The plates get coated, they get washed and blocked and stabilized. The plates that got finished for today are sitting in the drying racks there now. At the end of that process, once they're dry, they'll get pouched, sealed, and ready to be kitted.
In order to test that the product was manufactured appropriately, we have in-process QC steps along the way. So you know that the right material was put on the slide, you know that it's reactive. Finally, at the end, we do statistical sampling. So we take all of these materials and we have to destroy it by reacting it with antibody-positive samples, antibody-negative samples, difficult samples. And what they're doing right now is reacting those samples with positive controls. We'll examine it under the fluorescence microscope, and if everything looks okay, it's released to the public. Anita is our quality control manager, and what are you looking at now?
This actually right now I'm looking at some tissue slides which are mouse kidney tissues, and what you see on the screen are ANA homogeneous, that is anti-nuclear antibody reactions.
Our goal has always been to be a leader in autoimmunity, and we've achieved that goal. We've also wanted to be an example of the healthy Western New York economy. Our intention is to be here. Our intention is to grow employment locally. We want to be a good corporate citizen, and we want to help in the diagnosis and proper treatment of a number of people with autoimmunity, whether in the U.S. or internationally.